Picture the flow of information. While we talk about the nervous system being electrical, there is electrical communication or electrical flow within the neuron. So how the information flows from dendrite to cell body to axon to the end of the axon is via an electrical signal. And it's measured in volts. So it's going to be voltage that's going to be driving this current. Right? So that's some terminology we'll talk about with physics, is that potential is a measure of the strength, volts, the strength of charge. The movement of the charge, the flow of the charge is current. Right? You can have all kinds of potential and volts. That's what a battery is, right? It stores it. But you don't use that potential energy until you get current, the flow of the electricity through something. Does that kind of make sense? So the flow of this electrical potential, if you will, the current, is going to be within a neuron, inside the neuron. It's actually a property of the neuron's cell membrane. So this electricity is going to flow, I don't want to say through, let's say along the cell membrane. So the electricity moves along the cell membrane. It is a property of the cell membrane. But this neuron ultimately has to communicate with another cell. Right? Now this other cell depends on where we are in this pathway. This other cell could be another neuron. The other cell could be an effector. If it's an effector, it's a muscle cell or a gland. If it's another neuron, it's another multipolar neuron. Okay? But this space here, right, this physical space here between them, if you remember, we called that the synapse, or it's actually called the synaptic cleft or the synaptic gap. So we have a presynaptic cell here, right? The neuron is the presynaptic cell. The electricity goes here, reaches the end of the membrane of the axon, and then there's a gap. And then there is a postsynaptic cell out here. Now, what is this postsynaptic cell? It depends. If this is a neuroeffector junction, this is a muscle cell or a gland. If this is just a neuron-neuron junction, this is a neuron. Okay? So it depends on what it is. But what's interesting is that, let's focus on this being neuron-neuron to stay within this realm of this electrical communication. You're going to have electrical communication in the presynaptic neuron, and then you also want electrical communication in the postsynaptic neuron, right? We want that electricity to be able to flow. But we've got a physical space here across which that can't flow. Now, if these two membranes touched each other, right, remember I said the electrical potential is a property of the membrane. If these two membranes touched each other and were held together by like a gap junction or something like that, right? Like this would be analogous to, it's called a gap junction, but it'd be analogous to something you might, remember intercalated discs in muscle cells? This is what they look like. Those muscle cells are held together. So this right here, electrical signal right here, but because the two membranes are touching each other and held together, it just keeps going. So there's no break in it, right? So this is called an electrical synapse. When our brains and bodies are developing in utero, as embryos and fetuses, we have a lot of electrical synapses. As adults, or even as an infant and a child by the time they're born, we have very, very few of these. And the reason is simply because it's too rigid, right? If they're physically stuck together, it's a dedicated circuit. I can't break it and use this neuron for something else. This ability to break and reform connections is called plasticity. Do you think that's important in your, just think in your brain, is it important in your brain to be able to use neurons for different things? Yeah, we got a finite number of them, right? And we form pathways and we keep them intact while we're using them, but when we don't need them anymore, they're gone. And I use the neuron to do something else. So if I had all of these, they're dedicated. I can't use them to do multiple things. These are much more difficult to break and reform connections with. So rather than having these dedicated physical electrical synapses like this, where it's a rigid structure, we separate the cells by a space. There's nothing holding them together. So is this easy to break? Yeah, there's nothing holding it together. Just move it apart, go do something else. Okay. That's plasticity. Being plastic means moldable. The problem is now this. This was really easy to go electrical because there was no break in communication. Here, however, we want to go electrical, but this space is too big to spark that charge across. It's not like a spark plug where you can actually shoot the spark across. It's too big, right? The reason we can't shoot the little spark across is because the strength of this current is not strong enough. So what does that mean? That means I have to go electrical, and then this electrical synapse right here, at this electrical stimulus right here, at this point, is going to cause the release of a chemical. And then that chemical is going to get released right here and just diffuse across that. It's going to be a high concentration here where it's released, right? Low over here, so it'll just move by diffusion. And then that chemical is going to interact with the other cell over here to cause this cell to do what? Generate an electrical signal. So electrical signal, chemical signal, electrical signal. This is now slower, right? Because here, this is fast. This is slower. Okay. And this means that we have to have the electrical signal release a chemical. Now, if this is a neuron and it's releasing a chemical signal, what do we call it? Neurotransmitter. And if it's a neurotransmitter here that's being released, it is going to affect this target over here. There are I'm trying to think of a hydrophobic neurotransmitter. There are some gases that are kind of hydrophobic that are neurotransmitters, but almost all of them are hydrophilic. So that tells you there needs to be a receptor, right? And where is the receptor going to be on this cell? On the membrane. Right there. So now again, we can't get away from this. We have a protein-ligand interaction that we have to consider here also. This is no different than the stories we've been telling about hormones and signals and whatever. This could be what? This could be linked to an ion channel. This could be linked to an enzyme. This could be linked to a second messenger system. There are examples of all of those with specific neurotransmitters. And even with a specific neurotransmitter, something like acetylcholine, which is the most abundant neurotransmitter in the body, there are examples where it's linked to GPCR, so it triggers a second messenger.